Learning Objective 11 is about age-related changes in taste sensitivity. In general, our sensitivity to the special senses tends to decline with age. When it comes to gustatory sensitivity, this particularly occurs after the age of 60 and can do so for a number of reasons. The number of taste buds themselves decrease in the mouth. The remaining taste buds can shrink in size, and overall, the mouth produces less saliva, which can affect the taste of things. In addition, with olfactory sensitivity, this particularly declines after age 70 and can do so for a couple of reasons. Decreased, again, mucus production, like saliva in the mouth, decreased mucus production in the nasal cavity. And then the fibers and number of receptors for the odorant receptor neurons decrease with age. Now, in addition to age-related decreases in sensitivity, this process can be exacerbated by exogenous causes. Medications, diseases, smoking, exposure to pollutants and toxins in the air can exacerbate or accelerate the decrease in sensitivity to taste and smell, particularly the longer you are exposed to them. And this is important because if you have a decreased ability to smell and taste, yet you're going to want to compensate somehow so that you can continue to satiate those senses through the eating and smelling experiences. So an obvious index of this decline is the tendency that adults and those over the ages of 60 and 70 tend to add a lot more salt and flavorings, such as spices, to their food than children do, and they're just compensating for that loss of sense of taste and smell. And in particular, this has health impacts when it comes to individuals with hypertension and other body fluid or electrolyte problems, because the additional additives of salt to their food to compensate for the lack of flavor can overload the body with too much salt and contribute to hypertension. While we have sugar substitutes, we have yet to develop a safe and effective table salt substitute, although I believe that food companies are trying to do so, but it just isn't yet produced. Furthermore, loss of taste and smell not only can result in the need to compensate by adding to foods, it can also lead to further appetite suppression, which can contribute to weight loss, malnutrition, changes in immunity, depression, and deterioration in other medical conditions because of the suppressed appetite, because it simply just isn't as enjoyable as it once was. This is why an involvement in a dietitian and as a part of the healthcare team, for particularly individuals over the ages of 60 and 70, and anyone with something like hypertension or depression, malnutrition, anything like that, is going to be important because they're going to be able to counsel the patient in healthy approaches to increasing taste and smell, aroma experiences in order to promote healthy eating habits. Learning Objective 12 is about taste processing in children. Sensitivity to gustatory and olfactory cues are higher in childhood compared to adulthood, and in fact, the sense of taste and smell begin in utero. It is believed widely that this purpose is to prime the fetus to the taste and the smell, you could say as well, of amniotic fluid, which reflects the composition of the maternal diet in the surrounding world. Because the maternal diet would therefore represent the external environment to which the fetus would eventually be exposed, and would also prime it for the future foods it would likely consume based off of regional environments that the mother or the maternal diet represents. In addition, the taste of the amniotic fluid also primes for the rooting and the smell of breast milk after birth through this heightened smell and taste sensitivity. In addition, newborns respond to sweet tastes And it has been shown that they will respond to a sweet taste even immediately after birth. And neural imaging studies have shown that even a couple of days old infants will respond in a neural imaging study to sweet solutions that is similar to other stimulation that would presumably be pleasurable. This has been best utilized and studied in regards to the ability of sweet taste to act as an analgesic in infants and children during minor but otherwise painful procedures. However, this effect is best for more mild and minor procedures and not particularly during intense pain or prolonged procedures. But a small amount of a sweet solution that is usually a sucrose type of solution placed on the tongue of a crying newborn will exert a rapid calming effect that has been monitored through not only facial relaxation features, but physiologic changes such as decreases in heart rate that will persist for several minutes. And this is an example down here on the left of Sweet Ease, which is a kind of a sucrose solution that is provided in hospitals that work a lot with pediatric patients, and an example of a pacifier that you would just dip into the sugar solution and then give to the infant to suckle. The effects shown here is indeed transmitted by a sweetness in the oral cavity and the taste buds, because directly loading the stomach with sucrose does not exert the same sort of analgesic and relaxation features. And for this purpose, sucrose is generally widely used to help reduce procedural pain, particularly in single painful events such as heel lance and venipuncture and other procedures that are of short-term duration. And here is another example of a suckling of a sweet solution dipped into this sample provided here. I know this works. I used it myself when my son was in the hospital when he was about 4.5 or 5 months old. And during the times that they had to do blood draws or give him an IV, we would give him a pacifier that was dipped in sugar to suckle on. And he had otherwise always rejected pacifiers. And I had tried to get him to take one, but he would never take it. He always spit them out. But dipping it in that sucrose solution would immediately calm him and stop crying, and he would stop that kind of painful wiggling around and he would relax. And it definitely worked. The mechanism by which the analgesic effect of sweet solutions are thought to arise from a couple of different mechanisms. The most prominent mechanism is the anti-nociceptive action. We'll get to that in a second. So first, there is a proposal that just general brain arousal is suppressed because of the sweet solution, which could lead the infant to being distracted from what's going on. There might be a rewarding effect, in fact there probably is, to sweet flavors, which blunts a stress response, and so a decreased stress response can lead to a calmer affect. But the most prominently believed mechanism of the analgesic effect of sweet solutions in infants is through the anti-nociceptive action. Particularly, that sweet taste-induced mechanism results in a release of beta endorphins, which can activate the endogenous opioid system and reduce pain. In fact, sucrose administration that's been given to postnatal rats, meaning newborn rats, it's been shown to activate descending pain modulation centers. In addition, such as the periaqueductal Gray and Raphe nuclei that we talked about in your pain physiology lectures. However, these areas are generally believed to be quite immature and not very fully functional in young human infants. And so that's why the predominant mechanism that's believed to be at play here is in fact the endogenous opioid system activation through sweet taste-induced beta endorphin release. Now, in infancy and childhood, the preference for sweetness is quite high, and as we age, there is a prominent decline in preference for sweetness, and that is distinct from the decline we notice in olfactory and gustatory sensitivity in individuals over 60 and 70 years of age. Even the growing child into young adult and adulthood has a slowly generally declined preference for sweetness. Now, it's believed that this preference for sweet, very sweet things is linked to the child's need for calories, because we would think that a sweet item would be very nutrient dense and therefore provide calories for a growing child. However, since we have a lot of foods in our modern world that are very highly sweet but don't necessarily pair with high sources of other healthy and nutrition components, vitamins and minerals and things like that, the brain is actually physiologically unlinked between sweetness and health, or sweetness and energy content. So it becomes physiologically unreliable for the brain to pair those two things together. And this is a major change in human physiology in our modern worlds. And in fact, it's so recent to have these non-nutritive sweeteners and other food sources that are not providing lots of nutrients, yet are completely or very, very highly sweet. The long-term effects of this biological unpredictability, we can call it, is really unknown. It's going to take decades and possibly hundreds of years before we really know what ends up happening here. And I like this cartoon because it speaks very much to my life as a mother and my child, because he knows if there is anything quote unquote yucky in any ingredient that I give him. Learning objective 13 is about brain processing of taste. We're going to begin by defining the gustatory cortex. Again, this is a physiologic term, the gustatory cortex. It actually is made up of 3 cortical areas in the postcentral gyrus, frontal operculum, and the insula. And together, these 3 areas correspond to what we physiologically, rather than anatomically, call the gustatory cortex. Now, taste buds originate in this graphic down here at the bottom, and is going to involve the various nuclei of termination from their primary sensory afferent innervation. It ascends and then synapses in the ventral posteromedial nucleus of the thalamus, and then on to the gustatory cortex. This is a big picture of the gustatory input and kind of the processing all the way up to the gustatory cortex. The graphic begins with, down here in the bottom left corner, that the perception of flavor requires input from the taste buds of the anterior 2/3 of the tongue, posterior 1/3 of the tongue, and then the epiglottis itself can also provide taste input. In order to perceive flavor, you have to have gustatory input, of course, and you also require olfactory input. There's also somatosensory information from the mouth as well. For any of you who dislike the taste of a food because of the texture, that is relayed through somatosensory input from the oral cavity. And all 3 of these factors come together in an area called the medial orbital frontal cortex. So all of these are required to have the perception of flavor. And we're going to go through each one of these on its own. After the oral cavity and tongue, this information converges into the nucleus tractus solitarius, which also converges with various other branches of the vagus nerve. This relays, in addition, information about the oral cavity and taste, but also relays information about visceral activity as well. So this is an early site of gustatory visceral integration. Next, the ventral posteromedial nucleus of the thalamus is, like other thalamic nuclei, a relay station for taste perception. This begins the discriminative aspects of taste. And then it relays information onto the gustatory cortex, I showed you a couple of slides ago, which then is able to discriminate the various aspects of taste. Connections between the gustatory cortex and the orbital frontal cortex is allowing an individual to integrate not only taste, but integrating that with visual awareness of what food looks like, somatosensory information from the mouth and otherwise, olfactory input, along with taste, all converges again on the orbital frontal cortex for the entire kind of eating and taste experience. The affective aspects of eating, or providing emotional context to eating, memories of eating, things like that, takes place in the amygdala, which receives information from the gustatory cortex. In addition, the hypothalamus, which is closely related to the amygdala, integrates homeostatic mechanisms of eating or physiologic mechanisms of eating, such as hunger. That connection works together with the nucleus tractus solitarius. Working together, the limbic system, which also comprises of the amygdala, working with the hypothalamus, this modulates the interplay between eating and the kind of calming effect of food. This involves the limbic system as well as the reward systems, which we'll get to in the next lecture on reward and addiction physiology. And finally, there are medullary reflex arcs. So these are reflex arcs that just take place within the medulla and involves the nucleus tractus solitarius. And this forms the physiologic basis for things like salivating, mimetic responses, and swallowing. All, of course, are important components of the eating experience. Learning objective 14 is about brain processing of smell. First, we're going to look at the olfactory neuron system itself, and this particular connections at the earliest part of olfactory processing. It begins with the olfactory receptor cells, and we know that one olfactory neuron will express the odorant receptor on all of its cilia. So each individual olfactory neuron receptors, of which there's about 350 in humans, meaning there are about 350 different types of olfactory neurons, even if there are about 12 million individual neurons altogether. Now, a given olfactory receptor neuron, in this case, this blue one here, that expresses a very specific olfactory receptor, they converge, and their axons will converge onto one single glomerular layer, one single glomerulus, which is in this case located here. And there's a synapse. So the projections of similar olfactory receptor neuron cells will converge onto the same glomerulus. And then all of the olfactory receptor cells that express a different receptor type will converge onto separate glomeruli, which is located here. Now, the input from these glomeruli travel up to the next cell type. In this case, we have a mitral or a tufted cell, which is ascending on up to the brain, which is located here. Now, you'll notice there are other cell types involved in this process that I'm not going to go over at this time, but I'm sure that it is covered in anatomy. The olfactory bulb is kind of the next layer that we're talking about. We have our odorant receptor neurons down here, our basal cells or the support cells that are alongside them in the olfactory mucosa. They pass through the holes in the cribriform plate, and then they end up in their respective olfactory glomerulus. The mitral cell then projects its axons onto onward through the olfactory bulb and towards informing the olfactory nerve. Other subtypes of note are the granule cells and the periglomerular cells, which I don't really go into at this time. This is a kind of summary of the different layers in the olfactory processing in the olfactory bulb. Olfactory neurons that synapse onto glomeruli utilize glutamate. The mitral cells that are formed will project onto the olfactory tract. And then here are the other two cell types that are involved, I mentioned previously, the periglomerular cells and the granular cells. Essentially, they act as interneurons and utilize an inhibitory neurotransmitter, GABA, to inhibit kind of surrounding regions to help increase the specificity of the signal so that it can be relayed on through the olfactory tract. And from the olfactory bulb into the olfactory tract, we're going to project to the olfactory cortex. Just like other special senses, we have a primary olfactory cortex. However, like the others, this is the only sensory system that is unique in that it does not route through the thalamus before connecting to the cortex. So there is no relay station in the thalamus with the olfactory system. The olfactory cortex consists of several different areas, and similar to the gustatory cortex, that is kind of a physiologic term rather than an anatomical term. Here are the anatomical regions that consist of the olfactory cortex. They are the anterior olfactory nucleus, olfactory tubercle, piriform cortex, anterior cortical amygdaloid nuclei, periamygdaloid cortex, and lateral entorhinal cortex. And then from this wide olfactory cortex, we can have projections from this region to a variety of different regions. There are intrinsic projections that kind of stay intracortically, you could say. It stays within the cortex itself. It can project back to the olfactory bulb. There are projections onto the thalamus. So remember, this is the only sensory component that does not route through the thalamus on its way to the cortex, but instead the cortex routes out to the thalamus. Can route to the orbital frontal cortex, as we discussed in the integration of taste and smell earlier on with gustatory processing. Can route to the lateral hypothalamus or to the hippocampus. And next we'll go through each of the functions of this in turn. First, we have the anterior olfactory nucleus. This is a relay station for kind of going between the same side or other side of the brain. Generally, it's poorly understood other than being known as a relay station. Next is the piriform cortex, which projects particularly over to the lateral hypothalamus. This is involved in control of appetite because olfactory input influences appetite and hunger. The piriform cortex, as it routes to the thalamus and on to the medial orbitofrontal cortex, integrates taste, sight, and smell all together in order to allow one to appreciate the flavor and food experience. The anterior cortical amygdaloid nucleus is involved in emotional learning like other amygdala functions. And like other amygdala functions, it is involved in fear conditioning, particularly as it relates to olfactory input. The periamygdaloid cortex is involved in the integration of the emotional aspect of food as elicited by its smell. And then finally, the entorhinal cortex, as it projects to the hippocampus, is important in memory formation. Of course, the hippocampus is very important in the formation and recall of memory. Olfactory input facilitates memory formation and recall, and in fact, a memory that is paired with olfactory input will form a stronger synapse or a stronger memory tract. Connections within the limbic system and the entorhinal cortex are responsible for that, what we call a highly evocative experience of memory upon odor sensation. And I'll give you an example. Personally, I have this memory that comes up every time I smell warm, freshly laid asphalt, like what you would lay down on a street, really black asphalt. And the memory that pops up for me is a memory of childhood being at a theme park, because theme parks they always had those black asphalt kind of walkways and streets that you walk through to get to a theme park. And we always went in the very hottest part of summer, and that asphalt would just get baking hot and it would just smell like freshly laid black tar. And so every time, even if it's just walking down the street and someone is laying a new driveway going up to their house. When I smell that, I immediately think of being a child and going to a theme park.